for the recording there for everybody. All right, good morning. I'm glad you've given up part of your Saturday morning with us to go over enzymes and then the process of protein synthesis and the process of the central dogma that we'll talk about. I'm Robert Bauer. I teach at Barren County. I teach AP Biology and Biomedical Sciences. So I'll be with you for both sessions today. If you have any questions, feel free to just throw them in the chat and I'll get to them. If I don't, just throw them back in there again to make sure you get all your questions answered. So we'll get started. Make sure you understand this is College Board material, but we're not necessarily affiliated, and College Board does not endorse Advanced Kentucky as a product. So I assume this is mostly Kentucky people, but it could be others that are with us as well. Mr. Akampora is not with us today, but he's your science director. And if you have any questions, you can email Lou at that email and follow Advanced Kentucky on Twitter and Facebook for any updates. Make sure your microphones are off, your video's off, and your chat is the way we communicate today. I'm going to ask you to private message me some things in the chat, and I'll try to monitor that when I do. I'll also use the polling feature, and you'll just put in A, B, C, D, or E as your answer, and we'll discuss some multiple choice questions. So that's the way these couple hours will go. I'll try to go about 55 minutes or so. We'll take a 5-minute break at the end of this session, and we'll start the other session about 5 minutes late. So that'll give you a 10-minute break in between sessions. All right. Any questions before we get started here? All right. If not, we'll go ahead and get started with enzymes. So enzymes is our first session today. This should be review for most everybody. You would have looked at this in Unit 3, so might have been fairly recently for some of you, but it should always be review. So if you have any comp books, notes, whatever you've got, get those out and be ready to go. It's going to be more beneficial if you obviously put your phones up and you have your notes out and you use this as a beneficial hour or two. All right, so I do have the recording on, and I know a lot of people are taking the ACT today, so this will be recorded for everybody to go back and watch a little bit later. All right, so we'll start with some properties of enzymes. I've got a couple of things on the board, or slide, if you will, that you may be of interest to you. So what I'm going to try to do is in these boxes, as I call it with my kids, in these boxes, that is what comes directly from the curriculum that AP gives us. So this is what they expect you to know about enzymes. And if you know that, I think you have a good baseline of what you have to apply in those multiple choice and free response questions. So there's about 5 things that we want to hit to serve as a baseline for enzymes. Enzymes are biological catalysts. So what that means is they are a protein that starts and speeds up those reactions. So they are, as you see there, proteins. Proteins are going to help and have a specific shape. So proteins, they're biological catalysts. So that means they speed up the reaction. We'll get into how they speed up the reaction in just a second. And then a lot of times a common nomenclature is they end in ACE. Not all of them end in ACE, but usually if you see the suffix ACE, you can pretty much bet that you have an enzyme. And that the suffix is ACE, but the prefix usually refers to the enzyme substrate. So that's a pretty good indication. You know what substrate that enzyme is working on and that you have an enzyme and a protein all in the same. So what we're going to work on and look at is in these boxes. In addition to that, hopefully you're able to understand the enzyme includes an active site that's going to match up specifically with the substrate. So the interaction of the enzyme is going to take place within its active site. The active site is where that substrate binds, and I've got a picture of that in just a second. So this is going to make an enzyme-mediated chemical reaction. So the shape of that substrate and the shape of that enzyme have to be compatible, specifically the active site of the enzyme. So let's start with this first question in a private message to me, please. I've given you 3 properties of enzymes. They're catalysts. They usually ends in ACE. They usually end in ACE, and they are proteins. So based on box number 2, what I just read to you and what you can read again if you need to, what would be another property of enzymes? Put that as a private message to me in the chat, please. So they have to have a specific charge of the substrate. It has to be a specific shape, and that shape must be compatible with the active site of the enzyme. So I'm asking you for another property of enzymes based on box number 2, please. Everybody can use that chat feature for me, please. Okay, so this is another one that I haven't given you. So Kaylee, that's correct. We've got a lot of content to get through. So if you think you got a guess, throw it in there. Otherwise, we'll try to move on. Okay, Brady, good. So a couple of people have said that. Megan, that's correct. The shape of the enzyme and the substrate, the shape of the enzyme and substrate have to be compatible with each other. So the enzymes are specific. So that's your 4th characteristic. They're biological catalysts. They end in ACE. They're proteins, and they are specific. Enzymes are specific for the substrate that they will work on. All right. So let's go ahead and look a little bit about that specificity. So we have 2 different models here. This one on the left is what we'll call the induced fit model, and the one on the right is more of a lock and key. So let's look on the one on the right to see what we actually have. So this is the enzyme in purple, and then the arrow points to the active site. So the substrate has to fit into or be compatible with that shape. So you see these reactants come in, those reactants come in, and they are compatible with this kind of lock that you have. They fit the key. So that's why this is called a lock and key model. So the enzyme is the lock, the reactants or the substrates are the key. They fit perfectly. So we have an enzyme mediated reaction take place here. And ultimately, we're going to spit out some products. Okay. Because we can go around in a cycle here, and that enzyme is not used up in the reaction, that gives us another property of enzymes. And that's going to be our 5th one that we would come up with. Can anybody throw that one in there? Because we can go around and around and around. We say enzymes are this. All right. A little bit quicker there on that one for some of us. Thank you. So you could say they're recycled, or you could say they're reusable. So enzymes are not used up in the reaction. So enzymes are reusable. So to review, I've got 5 for you. They're catalysts, they're proteins. That means they have a specific tertiary shape that makes them functional. They end in ACE, they are specific, and they are reusable. All right, this is kind of the old school lock and key model. What we'll look at more is the induced fit. So what induces the fit? The substrate is going to come in again to that active site and induce the enzyme to kind of change shapes. So this is a big word that we're going to say here. It gives it, when that substrate comes in, it gives it its conformational shape. You see that a ton in essays, and they want you to say that specifically. So the substrate induces a conformational shape change in which we are going to get a reaction to occur. And then this is the enzyme substrate complex, but we don't have a C where it would show that kind of spitting out the product and being reused over and over. Okay. Again, if you have questions as we go along, just stop me, throw it in the chat. This is just pretty basic information about enzymes to get started before we actually apply it right here. Okay. I'm going to throw up a chat, or I'm sorry, not a chat. I'm going to throw up a poll. So this question should be just an easy one to get us started based on some properties of enzymes. So let me launch that poll. And feel free to put your answer in. I'm not going to give you very long on these. All right. We need to be trained how quickly we can answer some of these. Okay, about 10 more seconds is all we can give. So 5, 4, 3, 2, I'm going to end that now. And you see everybody pretty much nailed this one. We'll share the results with you. C is the correct answer there. And I'm not sure we even really need to talk about it, but that does definitely match up. What explains that the reaction specificity of an enzyme, it's that they are very compatible with the substrate. So very good. You guys nailed that one. So you definitely know that property of enzymes, and we can move on. So you see the box again. Enzymes have a regulatory function in biological processes. So I've told you, I've told you that they are biological catalysts and they facilitate that reaction. How do they specifically facilitate that reaction is what we need to focus on now. So if they're catalysts, that means they speed up the reaction. Let's look at this graph. This black line is the course of a reaction without an enzyme. So this looks like we start here with the reactants, we end here with the product. So that means the delta G is going to go down. That means that this reaction is spontaneous. This is an exothermic catabolic reaction, so it's releasing energy. You should be able to say all those things to describe that graph. But initially, it looks like we have to input some energy. So we're inputting energy to get that reaction started. Can somebody tell me what that energy is, please? What is that energy that we have to put in to get the reaction started? Private message, that one. Should be a quick throw in there. Maybe not what the energy is, but more specifically what that energy is called. How about that? A lot of us are throwing ATP in. That's not necessarily exactly what I'm looking for. So this EA here, it's a little, it makes it think a little backwards. That's activation energy or energy of activation. So we actually have to put a little bit of energy in. So that's the investment phase. That's the energy we're putting in to actually get this amount of energy out. So we put a little in, but we get a lot more out to make that reaction happen. So what enzymes do, enzymes are going to lower that activation energy. So specifically, they make everything a little bit more compatible. They help the enzyme and the substrate find each other. So if they find each other quicker, it lowers that activation energy and it makes this reaction all happen faster. So what you ultimately have to be able to tell me is we use this much less energy. If we use that much less energy, I'm sorry, I shouldn't have clicked it, but we use this much less energy. That means the reaction is going to happen faster. We save energy on the backside, and we ultimately get that product faster. Okay. So again, they're catalysts, and now we know that they speed up reactions, and the way they do that is by lowering that activation energy. Okay. So let's look at another question, as you saw on slide 11 here. And I'll launch the poll and give you about 40, 45 seconds to go with this one. Hopefully you do as well as you did on the last one. You can move the poll around if it covers the question up. About 15 seconds. A little bit more disparity here. A little bit more disparity here. Okay, so we got a minute on that one, so I'm going to end that. I'm sorry I didn't give you much notice, but we ended that. So the correct answer is the one that most of us went with there, and that is B. So we might need to look at it. We were all over the board there. We had about 6 right and 6 wrong if you look at that. So why? What statement best explains how the enzyme speeds up the reaction? So we're looking for We're looking for something about stabilizing that transition state. We're looking about lowering the activation energy. We're looking for something that matches up the properties that we just had. B meets that parameter the best. So the enzyme binds and stabilizes that transition state. So that quicker stabilization allows that reaction to happen, and it goes with a decrease in the activation energy. Okay. It doesn't really have anything to do with the amount of energy released by the reaction. It doesn't have anything to do with a decrease in the free energy change. Okay. So it's all about that activation energy. That's why it is speeding up the reaction. That's why we can call it a catalyst. If you're a catalyst, you're helping a process take place. That's just an everyday vocabulary word that you would hopefully know. All right, let's move forward. All right, so we have two different types of inhibition. You should have maybe learned about both of these, competitive and non-competitive. If you look at the graph, you can see what the difference is. So this is normal in A. That substrate binds to the active site, and we get a nice little product out of that. But if we have a competitive inhibitor sets in that active site. So if we set in the active site, clearly that substrate can't bind. We get no reaction. We get no product. A non-competitive inhibitor, they're not directly competing together. So if they're not directly competing together, then that would mean it's not really going into the active site, but it's doing something to that active site that causes this substrate not to bind. So you see in this normal phase, it looks like this, but this non-competitive inhibitor has bound to another site or bound to another site. So another site is called an allosteric site. So this is also known as an allosteric inhibitor. So if it's an allosteric inhibitor, we have this no longer able to bind because it doesn't really fit. Okay. So let's talk about two things real quick. Okay. In competitive inhibition, this is just something that's not on the slide, but we'll go with it. The substrate concentration matters here. Okay. If I have a ton of substrate, the green, if I have a ton of substrate, I can overwhelm that competitive inhibitor and actually bind to more sites and still get a reaction. So in competitive inhibition, the substrate concentration can actually cause the reaction still to happen if we have more substrates than we do competitive inhibitors, because they're going to bind to that site more because there's more than likely more than one enzyme here. Okay. In non-competitive inhibition, it doesn't matter. All right. The substrate concentration does not matter because we're not directly competing for that site. So we could have all kinds of these. They're still not going to be able to fit in or compete with. So you think about here in a competition, if I have more substrates, then I'm probably going to outcompete my competitive inhibitors. But here, if it's not a competition, it doesn't matter how many I have. Therefore, the concentration doesn't matter. This allosteric guy isn't directly competing with the same site, so it's still going to change that site and negatively affect you. Okay, hopefully that made sense. And if it didn't, just stop me and say you're not making sense, and I'll go back and try to explain it another way. Okay. Ultimately, we're here to try to benefit you guys and take something from this that's going to positively affect you on May 14th or whenever you decide to take the test. I know there's a couple options this year. Right. So let's see a little bit more detailed question about inhibition, since that's what we just went over. I'll launch the poll after you've gotten a second to read it, maybe. Okay. We've gotten about the majority of us come in there, so I'll stop that in about 5 seconds. And it looks like, if I get those lines off there, it looks like most of us were paying attention and came in with the right answer there. So I've annotated a few things there that I think were important to help you answer that question. You can look at that, and it's okay if we're not graded necessarily on how well we do. I just want you to learn from your mistakes if you got it wrong and do better, okay? When it really counts on test or ultimately on the exam day. So we know we've got an enzyme. It ends in ase. It tells us that, though. It breaks down ethyl alcohol. We have a competitive inhibitor. So we know that competitive inhibitor is going to compete for that same active site. So it's kind of backing up everything we know. And if you know this stuff, though, it allows us questions to read easier, which is going to help with our time, which is going to ultimately make us feel more comfortable as we take the test. So we have a competitive inhibitor that does not allow non-toxic substances to be made. Instead, it's actually making more of a toxic substance in formaldehyde. So ultimately, the question asks, what's going to happen if I increase the concentration of the substrate? Well, what did I just tell you? I told you that if it's competitive, which we have matched up there, if it's competitive, the concentration matters. So let's look for one where it says that the concentration matters. And most of us came up with the right answer there, and we said that the answer was D, because the inhibition is going to decrease because the active sites occupied by the substrate is going to increase because we have more substrate. Okay, so very good connection there, and paying attention, following along. Very good. Okay, I'll erase all that there, and we will get going to the next little section. All right. So what I've thrown at you here is it was actually a multiple choice question at one point, but they've changed it into a free response. So we're going to have to take a second, and I'll put a timer on us. But what I'm going to do is I'm going to ask you to answer this part by part. So when we look at free response questions, we're going to have a graph, we're going to have some kind of reading, but we're going to have bold words. Those are our action verbs. So in this situation, this is going to be worth a couple of points. So I want you to predict first, and then we'll go over that, and then we'll support. Okay. So in the private message, I want you to throw in your prediction if we add a non-competitive inhibitor to the reaction mixture. Whenever you get that, just hit enter on the chat. So a prediction is just going to be a very simple sentence, a prediction of what will happen. And there's only a couple options here that you can ultimately pick. Okay, so this is the point where we've got to really work on our essays. We've got plenty of time left in the game to make sure that we're doing what we need to basically just answer the question as it's written. Okay, so what is the question asking and what do you have to address? Ultimately, you have to address the rate of the reaction. Okay, that's why I have that circled. So ultimately, you're predicting the rate of the reaction at a high substrate concentration if we had a competitive inhibitor. So I know I just completely rearranged the question, but to me, that makes more sense. So we are predicting the rate of the reaction when we have a high substrate concentration in the presence of a non-competitive inhibitor. Sorry, I put a timer on you for 2 minutes and most of you had that done a little bit quicker. Okay, so I went ahead and went. All right, so what is the rate of the reaction? I just told you that non-competitive inhibitors were substrate concentration independent, right? So no matter how much substrate concentration I have, that non-competitive inhibitor is still going to compete positively with the inhibitor. Or I shouldn't say compete, it's still going to inhibit the reaction. So your answer has to be that the rate of the reaction is going to go down at any substrate concentration. All right. Why is that the case? Because non-competitive inhibitors are substrate concentration independent. Okay. Does that make sense to everybody? The rate of the reaction will go down. All you have to say, period. You could add in there that it's going to go down at no matter what the concentration of the substrate is, because those two things are independent of each other. Okay, so what am I testing you on? I'm testing you on that last slide to understand the difference between one of them being concentration dependent and the other one not being. Okay, so let's take part two. Let's take part two. So I've given you part one, basically. So see if we can articulate what we just kind of went over. Support your prediction. Support that prediction. Let's say we all said it's going down. Let's support why it's going down. So let's see if you are retaining what I'm kind of trying to get at you here. Why will it go down? Again, private message me that, please. So remember, it's a non-competitive inhibitor. It's not a competitive inhibitor. But the rest of that looks good. All right, Nathan, good. So Shirley, make sure you're telling me why it's not going to work, right? Why is that reaction going down? You've already told me the reaction's going down. So why is the reaction going down ultimately? So think about what that non-competitive inhibitor is doing to cause the reaction to go down. Right? We've already said it's a non-competitive inhibitor. We've already said it's going to work no matter what the substrate concentration is. So ultimately, what is the reason that it's going to go down? All right. That's Brittany, that's a good answer. All right. So ultimately, we've got to understand that the substrate cannot bind to the active site. So because that non-competitive inhibitor is bound to that allosteric site, it's still going to change the active site. So there's no binding between the substrate and enzyme. If there's no binding between the substrate and enzyme, we can't get a product to form. So some of you are giving some really good answers. Some of you are giving some really good answers there towards the end. Okay. All right. So we're about halfway through. And let's go back to kind of change gears here. So this kind of concludes just the overall basis of of what enzymes do. We've gone over the functions. It's a catalyst. It speeds up the reaction by lowering the activation energy. It's a protein. Mainly the tertiary shape of that protein allows for the specificity to match with the substrate and its active site. So it's specific. It ends in ACE. And we went over the induced fit versus lock and key. We talked about non-competitive and competitive inhibition. So now let's just look. Let me get that off so we can go forward here. So these are just a couple of examples. You see one ends in ACE and one doesn't. So Ribisco is a very prominent enzyme in photosynthesis. It helps make that sugar, carbon dioxide specifically, into the sugar. Topoisomerase is something we'll look at in the second section. It's involved in DNA replication. So there's a ton of enzymes they could ask you about. The more confident you are that you've heard some of those before, again, the easier you're going to read some of those questions, and it's just going to make everything go a little bit smoother for you. Okay. All right, so let's shift to one little aside. So this is not directly about enzymes. It is cellular respiration related, but in this section, they're going to make you calculate rates. All right. So I'm going to ask you to calculate the rate. You've got a computer, or you've got a calculator close, whether that's on your computer, your phone, wherever it is, or maybe you just have a calculator laying around in your pocket, but calculate the rate of oxygen consumption in the germinating peas at 26 degrees. So that's going to be this line up here, obviously. We've got oxygen consumption here. We've got time here. So the ultimate question is, what is the rate formula? What is the rate formula that we have to know to be able to use, whether it's in enzymes or whether it's just in a normal graph they give you about just about anything? You may know what that rate formula is. Not the answer, just the rate formula. I'll throw the poll up in just a second. So you could say rise over run. That's one way to say it. You could say change in Y over change in X, or you could say change over time, since time is always going to be that x-axis. Okay, so those are three different ways to say it. So it's also somewhere that you're going to have access to, and that's on your formula sheet. So the more familiar you are with your formula sheet, the better you're going to be when you have to quickly answer one of these questions. Okay, I'll throw that poll up now, and let's see if we can use that formula, change over time, to actually get this right. So apply that change over time, change over Y over change in X to get this correct. Okay. We're about 60% of you voted there, so I'm going to end in about 5 seconds. Okay. 1, 2, 3, 4, 5. All right. So most of us have picked A, 13-to-4 ratio there. So that's about 75% of us getting it right. So how do we do this if we're confused? So I drew a poor line there, but this is going to account for my change in Y. So I just picked these 2 points here. It should be a constant slope going up. So 0.75 minus 0.25, that gives me 0.5 on the top. And then I'm going to pick those 2 times that I went to. Obviously 15 and 5 would give me a difference of 10 minutes. So it looks like I am, 0.5 divided by 10, which gives me that A number there. Okay. So if you're having trouble or you don't remember how to do much of that math, hopefully you can ask your teachers to kind of go over that. A math session, a review session would be a good session to have kind of at the end of when we got all of our content done. All right. So let's officially shift gears now and go to how environment influences enzymes. Okay. So you see our content is very long there, but ultimately environment's going to influence how that enzyme works. All right. There's two main things that we'll look at here in this picture, and that's pH and temperature. So if you look on B over here, environmental temperatures and pH outside an optimal range. Now, I find a lot of people honestly don't know what optimal means. Optimal is just a fancy word for best. So when it works the best, or when it works the highest, or when it's functioning as it should. So environmental temperatures outside the optimal range for a given enzyme will cause it to change its structure. We know if the enzyme changes its structure, it's ultimately going to alter that active site. If the active site's altered, the substrate can't bind, which means it is not going to work. So we have an optimal range, and then when we're outside of that optimal range caused by a change in temperature, change in pH, we don't get the enzyme to work the way it should. A fancy way to say that is the enzyme can be denatured. So that is a big fancy word for basically just saying we know it's a protein, and because it's a protein, when that protein denatures, it just unfolds. Okay. I told you earlier that it needs to be in its tertiary shape to be functional. If it unfolds, if the hydrogen bonds are broken, or if any of those bonds are broken that allow it to go back to its primary shape, which is just a bunch of amino acids connected together by peptide bonds, then it's ultimately not going to have an active site. If it doesn't have an active site, we can't bind, we can't change the substrate into a product. So please understand that environmental temperature and pH cause it to denature. It's not just enough a lot of times to say that it denatures. You need to say what that denaturation means. That it's just unfolding and no longer has its function. Okay, so let's look here. We have two different enzymes here. This is an optimal human enzyme. This is a body temperature at Celsius. So it's going to function clearly at body temperature the best. If we have a fever, or we are for some reason under that fever, our body temperature is lower, that optimal range is going to leave, and we're not going to get as much reaction. All right. If we switch and go to a thermophilic or heat tolerant bacteria, obviously it's going to want to operate in a much higher temperature. If we switch those, it's not going to work. If we move the human enzyme into 77 degrees Celsius, it's going to get denatured. It's going to unfold. Okay. The same way over here. All right. We have stomach enzymes and we have intestinal enzymes. All right. A lot of our digestion does happen in our stomach, and pepsin is one of those enzymes that's able to break down proteins, digest them, not break down our enzyme, but digest proteins. And trypsin is going to work in the intestines. So if I take trypsin and put it in the stomach, not going to work. Or if I take pepsin and put it in the small intestine, not going to work. So ultimately, they have their optimal pHs, which gives them their optimal location to operate. Okay. So if I change them, if I change their pH, their structure is going to be affected. If their structure is affected, problems. If I change their temperature, their structure is also affected. Okay. Most of the times, this is not a reversible process. There's very few times that it is actually reversible, allowing it to regain activity. But the majority of times when denaturing happens, it's hard for it to fold back up and be reversible. All right. So let's answer a question about that. So what I've done here is I've got three different enzymes: pepsin, chymotrypsin, and trypsin. And it's very hard to see because I had to get it all on here. So hopefully you can read about those three enzymes. Okay, I'll give you just a second and then we'll explain that. So I'll start the poll and leave it up for about a minute. Okay, so if you couldn't have read that up there at the top, we'll close it out in about 5 seconds. I tried to diagram that out. So the pepsin is going to be active in the stomach where the pH is about 2. The chymotrypsin and the trypsin are going to be active in the small intestines where the pH is about 8. So I'm going to close that out. We are a little all over the board on this one, unfortunately, but the majority of people did pick the right answer, which should be C there. So I'll share those results. Unfortunately, more people got that one wrong than right, but that's okay. Let's look and see why that happened. All right. So we've got the background information. This one's in the stomach. These are in the small intestines, which makes sense that they have the highest graph around 8. This one maybe works a little bit more basic. All right. And then ultimately the stomach is going to have the highest or optimal range about 2. Okay, so which of the following best predicts how the structure and function of pepsin will change as it enters the small intestine? So if you know anything about the digestive system, things in the stomach get entered into the duodenum or the first part of the small intestine, and they move further down those small intestine before they enter the large intestine. So the stomach is very acidic, but as far as we move away from the stomach, it looks like we have to get more towards the basic side even to get those enzymes to work. So your answer should be the pH shift as we move towards the small intestines. Okay, so if we move pepsin into the small intestine, obviously it's going to change shapes because of that basic environment in the small intestine. So that's why the stomach enzymes activity is going to decrease as it moves towards the small intestine. Okay, and hopefully that makes sense for you. Okay, let's see. All right. I'll get all that off there in just a second. All right, so what I've done here is I've actually taken a multiple choice question and I've made it into an essay type question. So what I need you to do is read that. I've got to move all my stuff here so I can even read it. Very simple answer. If the experiment is repeated at pH 11, what's going to happen based on maybe the trend here? What's going to be the most likely observed level of enzyme at pH 11? All right, private message me that one, please. This should be just a very quick. I mean, this is you don't even have to put it in sentence form if you don't want to. Okay, very good. All right, Addison, I like how you said that there. Put it in sentence form there. So at pH 11, the observed activity would be less than even pH 9. Okay, it would be less than pH 9. It would definitely have an optimal range, probably in that 5 range. But you're going to say that it's going to be less than the rest of these, okay? Because it looks like we have a decline. It should be the rest. I'm sorry, it should be less than the rest, okay? So very good there. And I think you would have got that multiple choice question right if I had left it that form. All right. All right, couple extra things here. So we have talked about pH and temperature. Now let's add something else to it called substrate concentration. I can also change the substrate concentration and the enzyme concentration. So pH, temperature definitely affect it, but other concentrations, substrate, enzyme, even cofactors, which are going to help, will definitely affect positively or negative how that enzyme works. So I put the boxes here that say that. So concentrations of substrates and products determine how efficiently an enzyme reaction proceeds. So if you think about it, substrates are going to be the work and the enzymes are going to be the workers. So I've got to have enough work for the workers to do in a concentration change is going to drastically affect how they actually work. Okay. So let's look at some examples in this situation. I'm going to put up some graphs here for you. All right. So we've talked about temperature. We've talked about enzyme activity. We've talked about the optimal rate. I'm just kind of reviewing real quick before I do. I'm sorry. So we need to know what optimal means. So optimal activity here would be 35. Optimal activity here would be 7. So the reason that that's optimal, this maybe would make sense to you before we move on. The reason that this is optimal is because higher temperatures are going to increase to some point the collisions. So once we increase the frequency of the collisions between the enzyme and substrate, that's a good thing because it's going to increase the rate of reaction. But at a point where it gets too high, it's going to ultimately denature that enzyme and that activity is going to drop. All right. So you see it affects it a little bit and then it kind of drops off where it gets too high to denature. The same thing's going to happen with the pH, right? Colder temperatures usually take smaller, or it takes a longer time to decline. You see the slope of that graph is shallower versus it just drops here. So colder temperatures usually affect it slower. A lot of times even colder temperatures don't denature it. It just slows those collisions down so far that we have almost no activity. Okay, now let's go. Okay, so this is the substrate concentration graph that I'll show you. Okay, so substrate concentration, the more substrate you have, again, you have more collisions. So you're going to have more collisions. Let's just look at the black line here. Substrate concentration, the more you have, the more collisions you have, until ultimately you reach this point right here known as the saturation point. So we are saturated with substrates. Every worker has work to do, and until they get that work done, they can't get more work, if that makes sense. So in this point right here, every enzyme would have work to do. We don't have any empty active sites. So ultimately that rate is going to drop off. We cannot increase it because we only have so many enzymes. All right. The change in the graph here, you're saying, well, why does this one not level off? Well, if you notice, what I did was I changed the y-axis. Okay. Here it's pressure. All right. Here it's reaction rate. So the reaction rate levels off at saturation when the saturation 1-to-1 level occurs, if you will. Here, the pressure, you see the blue line, the red lines go up. So we increase, we increase, we increase, we increase. So we're increasing the substrate here, a concentration at every line. But eventually we reach that saturation where we only can get so much pressure to build up because we, again, have that 1-to-1 ratio where they can't do any more work. Okay. Does that make sense for everybody? So more collisions until eventually saturation occurs and all active sites are occupied, right? Let's look at the opposite. So let's look at enzyme concentration. So enzyme concentration, again, I have the same graphs, pressure here, reaction rate here. As I add more enzymes, they're going to do more work faster. So I have the same amount of work, but I'm adding more workers so they can do the work faster. So this is just a regular rate here, but ultimately I like this one a little bit better. So let's say this is at 100% concentration. Maybe this would be 50 or 75, maybe. This would be 50, this would be 25. You see the pressure builds up slower. So let me just humor me here for a second. Let's see what happens. So would they all eventually reach this point of blue? My question is, would they all actually reach that pressure point? And the answer would be yes, but I don't have enough screen to even draw it out that far. What's going to happen is, let's see if I can do that quickly. So this line will eventually slowly move up, maybe even slower than that, but it's eventually going to reach that level. It's just going to take it a longer time to do that because we only have very few workers. We still have the same amount of work to do, but we don't have enough workers to do that. Here we're increasing the work, so it's going to get to that point quicker. Okay. It's going to get to that point quicker. Here it's even going to get there quicker because we have more workers. Okay. So does that make sense for everybody? We're going to get to that same pressure. We're going to get to this pressure point eventually, but it's taking a longer time for that work to be done because we have a smaller amount of workers. So in an enzyme sense, there are just a lot of substrate left over for those enzymes to have to cycle through their active sites. Okay. Hopefully Hopefully that makes sense. And feel free to throw a question in there anytime or something that doesn't make sense. You're not going to hurt my feelings at all. All right. And clear those off. Let's go to the next slide and we'll see if we can answer some questions here. Okay. So back to a multiple choice question there. We've talked about the substrate concentration. We talked about the enzyme concentration. So now let's answer a question about that substrate concentration. We've got about 5 minutes left. So about time for a couple questions, three questions, and then we'll call it break time. Let me find the poll and I'll throw that up. Again, you can move that poll around if you need to see things. This one should be a quicker one if I've made sense at all. Okay, thank you for participating. Most of you, as I said, have already got that answer in. Okay. All right, we are hovering around B. So let me share those results. We actually are 50-50, actually a little bit more than 50-50 split on this answer. So what's happened here? What's happened here is we have reached that saturation point. So the question asks, why did we have that change in slope at 3 to 5 minutes? Why did we reach that saturation point? So the top sentence basically just says we're measuring the product. Why did we stop measuring? Why did we stop getting product? Well, could we have run out of something? What would we have most likely run out of if we leveled off? So if we stop forming product, mainly the thing that we would have run out of is substrate. Maximum velocity, I'm not sure what that means really. An allosteric inhibitor could have appeared, but more than likely they just don't appear out of nowhere. The most likely explanation is usually the one that's the simplest. Okay, so the substrate's all used up. We're not going to get any more product. So that's why the answer is C. Let's try another one. Okay. So this will probably finish us out for this session. Everything I have left is our writing questions. So let's just finish with this one. Explain why a change in the reaction was observed after 30 minutes. So why was a change in the reaction observed after 30 minutes? This one has to be a private message, and then I'll give you about a 10-minute break. So we notice about 40 minutes, it starts to, maybe after 40 minutes, it starts to level off and it's not increasing very much. Okay, ready, good? Okay, so why is there not a change in maltose concentration? So maltose is the product that we're making. Why do we not make more maltose after about 40 minutes? Okay, I'll give you about 30 more seconds and then we'll go over that and take a break. Okay, so I think we're getting there. Okay. All right, so there's three possible explanations that they would actually accept in this situation. Again, the easiest one is usually the one that most of you have, or that's not the easiest one, but the simplest explanation. So that is that the majority of the substrate's actually used up. Okay, if there's no substrate, there's no way to make more product. So that's why you see it level off. So the first one that they would accept is the substrate's been used. Okay, the second one is maybe there's some type of inhibition there, but again, that's probably not as likely, and even less as likely as the enzyme actually loses its activity for some reason. But those are technically all three acceptable at equal levels. Okay. So I appreciate you hanging in there. If you have any questions, please feel free to use that chat. I'm going to pause my screen and pause everything here, and we're going to come back together. I'm going to give you about a 10-minute break. So let's just round it off and say we'll come back and start the next session at 10:05. Okay. If you have any questions, I'll fill those in the chat box, and you guys take a quick break, and we'll see you back at 10:05 for protein synthesis. All right. So this session is basically gene expression. Gene expression is taking our DNA, our genetic material, making it into a temporary copy of RNA that we can use as a template to make proteins at the ribosomes, otherwise coined the central dogma. So this is the kind of the central dogma of biology, how all of it works, because proteins, as we just talked about, like our enzymes, proteins are representing a large part of technically all of who we are. So this is a lot of information. And what I know about how this year has gone, for most of us, we haven't gotten there yet. So that means this is more like a preview than it is a review. If there's things that you don't understand, maybe jot it down, ask your teacher, or we can hang out just a second afterwards and hang in there and figure it out. Okay. So let's get going with protein synthesis. Okay. Appreciate all of you again being here. Hopefully the first session was beneficial, and maybe this one will be too. All right. Again, here's our boxes. This is our curriculum again. If you weren't with us the first session, most of you were. So we've got to understand that DNA is our hereditary information. That's the primary source of hereditary information. In humans, DNA is. In some cases, RNA is the hereditary information, but in most organisms, it's DNA. So genetic information is obviously transmitted from one generation to the next through the passing of that DNA. There's a little bit difference in prokaryotes and eukaryotes. This is something I make my kids know. Prokaryotes have to be a single circular chromosome, and eukaryotes, it would be multiple linear chromosomes, like you have 46 chromosomes, right? So plasmids is something that you'll learn about in the next unit, so I'm not going to touch on that real quick. But the main thing I wanted to touch on in this picture is this. So we should know, based on our first unit, that DNA is made up of nucleotides because it is as well as RNA, a nucleic acid. Okay. Nucleic acids are made up of nucleotides. Nucleotides we know have three things, and we see those three things in this picture up here. So they have phosphates, they have a sugar, and then they have a nitrogenous base. If it's DNA, that sugar will be deoxyribose. If it's RNA, that sugar will be ribose. That's one of the differences. Okay. RNA will also not be double-stranded, as you see here. RNA will also use uracil instead of thymine. So I know for certain this is DNA because it is double-stranded and it also has T's. All right. What about this down here at the bottom? It's talking about purines and pyrimidines. All right. Purines are double ring nitrogenous bases. Pyrimidines are single ring nitrogenous bases. So I know for certain that this is the T if this is the A, okay? Because A would be double ring, T would be single ring. So these are pyrimidines, C's, T's are all pyrimidines. So all single rings are pyrimidines. All double rings are purines, so A's and G's. Another difference between that pairing, or another way to, a characteristic of the pairing is what I should say. G's and C's have triple bonds. Those are hydrogen bonds that connect those bases. So G's and C's have 3 bonds, A's and T's have 2. So if I were to erase the T's and A's, the G's and C's, I should still be able to identify them because there's 2 bonds here. I know that's a double, so that's the A, and that's a single, that's a T, because there's 2 hydrogen bonds between A's and T's. Okay, and then the same process can be taken down and applied to the C's and G's. Okay, another misconception that people have is what are chromosomes? What is DNA? DNA in that loose stringy form, we know is chromatin when it's just hanging out, in the G phase, S phase, G2 phase of interphase. But when it starts to condense in prophase and metaphase, we're going to get this chromosome looking thing. This is actually 2 chromosomes because it's been replicated in S phase. So that's what you see. And the misconception is that this is what a chromosome is. Yes, it is. But this chromatid is actually a single. This is another chromosome. So this is 2 chromatids or a replicated chromosome. Hopefully you went over that when you talked about mitosis. Okay, so that part is a little review. The naming of the DNA may be a review too, because we should have done that in Unit 1. Okay, so let's get to the more complicated stuff. Let's start with a question. So I've given you a property. Let's see if you can make that property make sense in a multiple choice question. So let me find the poll. I will launch it, and let's see how quick we can answer this one. I don't think you want to see the results that quick. Let's relaunch the poll. Sorry. There we go. So a lot of words here. We got 4 responses and 4 different answers. So let's see where we got to go here. Hopefully it'll even out. Okay, starting to do that a little bit. So this is Chargaff's rule, and you read about Chargaff up there at the top. Chargaff said that we base pair A to T, G to C. So there's a lot of distractors in this question. And most of us have picked through those to show that the answer is B. Okay, so I'm just going to end that poll since I told you the answer. So what we got distracted by a little bit in this one, I understand. It says that there's approximately 50% A and G. Well, that would mean there's 50% C and T. That is not true. The DNA does not have to be proportional. Okay. So that would mean it's 25% A, G, C, and T, and that's not exactly correct. What is correct is the amount of A's and T's should be proportional. Well, the amount of C's and G's should be proportional as well. And that's what we see. So 17, 17, 33, 31, it's pretty close. But it's definitely proportional with the outside here, 29, 29, 27, 27, 22, 22, but it's definitely not 50-50. Okay, it's definitely not 50-50 in any of these organisms. Okay, so hopefully that makes sense to get us started. All right, so I had to throw a little bit about replication in. Replication is DNA to DNA. So we know replication as the S phase of mitosis. It's also the S phase of meiosis. Which you may be doing right about now. So how does the replication is what allows us to have the continuity from generation to generation. In mitosis, it's what allows us to have the passing of that DNA in meiosis. And here's some enzymes that we really want to look at. So this is just the basis of it. There's a ton of enzymes involved. You notice a lot of those enzymes end in ACE, as we saw earlier. So what we have is the DNA has to be synthesized. That basically just means we're making more DNA. So topoisomerase is going to come in, and since it's a double helix, it's wound up. Topoisomerase loosens that coil, the supercoil Helicase is going to unzip it, so it's breaking those hydrogen bonds, and the single-stranding binding proteins are like little bumpers. Those are bumpers to keep it from bonding back, because if we don't keep them apart, they're naturally just going to glue back together, and that helicase hasn't done any good. All right, primase is going to lay down my kind of template for where I need to start. So that's just an introduction to those enzymes, and that was quick, I know, but topoisomerase uncoils it, helicase unwinds it, and primase lays down, excuse me, my primer, so we can get this whole process started. All right, so let's look at it a little bit more in depth. Okay, there's two sides, and what I neglected to tell you was this, that you really need to know. New DNA has to be made in a 5-to-3 direction. That's the main rule, and you'll see that in transcription as well. DNA has to be made in a 5-to-3 direction. So what does that look like? That means we have to always jump on at the 3' end, okay? If we jump on at the 3' end, well, here's my unzipping, there was my primer, and this guy right here is going to be called DNA polymerase. DNA polymerase, it's an enzyme that's going to lay down my new DNA. So it's just going to use this template strand right here to lay down the new bases. DNA is what we call semi-conservative. So that means we conserve some of it. So what we use is, this is the dark blue is the old strand. Sorry. Dark blue is the old strand and light blue is the new strand. So it's conserving a template. So we're always going to get one old and one new. That's why it's semi-conservative, okay? So we're adding at the 3' end in a 5-to-3 direction. Because we can do that with DNA polymerase here, going straight down, we call this the leading strand. So we continuously add to the leading strand because it meets our rule. We're adding at the 3' end in a 5-to-3 direction. Well, you're saying, well, what about the other end? That's a 5' end. I can't add there. Can't add there. But the 3' end down here on this side, I can. So that's why we call this bottom strand the lagging strand. So we actually have to add in pieces. So this is hard to show on a non-moving, just 2D animation. But as this unzips this way, we have to actually add backwards because it's going to open up that strand. So we add in pieces. So there's my primer, and we can add the polymerase this way, right? So as this is going to the right, the unzipper or the helicase is going to the left and unzipping more. So then we can jump backwards, lay down primer, lay down new DNA, and just keep going over that process over and over. Since we're adding in fragments right here, that is called Okazaki. Those fragments are called Okazaki fragments. All right. Ultimately, we have to have two other pieces, and we'll show you those two pieces in the next slide. And as we look at it in kind of an overall picture, that makes it easier to see. So this is a continuous add. So as it's unzipped, we can have that polymerase come right behind and add it. This is a segmental add. So as it unzips to the left, we have to add in pieces, add in a piece, add in a piece, add in a piece. Well, we have DNA polymerase that comes in and removes the RNA primers, and then we have this enzyme ligase come in and zip it up, okay? So that's just the introduction to that. It actually happens in a bubble. It happens in a what we call a replication bubble. So we start right here where my arrow is, and we go to the left and we go to the right. We do that because it's quicker than just starting at one point and adding in one direction. So we're actually adding in two directions. Okay, now your question's like, where do these 3' and 5' ends come from? Well, if we go all the way back to the beginning, we notice this top end is labeled 3, 5, and this one's labeled 3. So that comes with the directionality. One thing that I kind of forgot to tell you at the beginning was DNA is anti-parallel. So it's parallel in the fact that it runs this way, anti meaning it's opposite end. So 5's up here, 3's in up here. So it's basically just which way the phosphate and the sugar is pointing. Okay, so that's how we show the directionality, and that's also what we use to know where to copy as far as the replication is concerned. Okay, just a very brief introduction. And in an hour, it's very hard to get through all this very in depth. So we're kind of touching on it because it is a preview for most of us. So we'll at least have seen it to know where we're going in the next unit. Okay, hold on, let me let somebody in here. Okay, all right, so I've got a question for you. And let's see if we can answer that one very quickly. Throw on a poll, and good luck. So this is just saying, which way are we adding here? Where are we adding? What's my adding rule? I gave it to you very quickly. Where do we add, and in what direction do we add? Okay. So I'm going to type this in. I see that a lot of us maybe didn't get that point, and I know I'm going fast. We add to the 3' end. Right? We add to the 3' end. So we can add continuously at the 3' end in a new, and so the new is added in a 5 to 3 direction. Okay, that might change some of our answers if it does. So we also know we absolutely cannot pick a couple of these. Which ones can we not pick? We can't pick C and we can't pick A because those show us adding continually in both directions. That's not possible. So those of us that pick those two, it's not possible because we said we had to only add at the 3' end. So let's see which one, where we can add. So we can only add here. We cannot add at that 5'. We can only add here. We can only add here. We can only add here in a continuous direction. So again, that also eliminates which one? It clearly eliminated A again, didn't it? So if we're adding the same direction, we need to figure out which way. Are we adding in the same direction, or are we adding in the opposite direction? So we've eliminated A and C, so is the answer B or D? It has to be which one? It has to be D, because we're adding in a 3 to 5 direction, or I'm sorry, we're adding at the 3' end in a 5 to 3 direction. So we're adding here at the 3' end in a 5 to 3 direction on the new. So your answer is D, and the majority of you got that right. Okay. All right, next, I think we have another question coming up here. So let's relaunch that poll. And let's go to that next question. Okay, I'll remove all my annotations while you read all that. So let's see if we learned from our last question. So how are the new nucleic acids being synthesized? How are the new nucleic acids being synthesized? About 10 more seconds. Five, four, three, two, one. Okay. So B was our right answer. Good job there. What in the world are all those lines that I put on there? The red lines was my original parent strand. So those are my template strands. And then the blue strands are my new. So it looks like I'm adding here at this 3' end, and I'm adding in a 5 to 3 direction. That's correct. I'm adding here at the 3' end in a 5 to 3 direction, and I'm doing it in segments. So that is my Okazaki fragment. So B is my right answer because the new ones are synthesized in a 5 to 3 direction. Okay, so that's just something you got to know. It's something you got to know, but you can also see it here in this that we are adding at the 3 in a 5 to 3 direction both ways. Okay, so these two questions, the previous question that we asked and this one, are really the same question, just asked in a very different way. Okay, let's move along. See if we got a question here real quick in the chat. Okay. All right. So this is just an infographic of an overview of what's going to happen in protein synthesis. All right. So we take our DNA template strand. So I've just zoomed in on a little piece of DNA. So this is a segment of gene 3. You know, we have DNA and the genes are on our DNA. So those genes code for different proteins. And we're going to make a segment of that protein from gene 3 here. So this is the DNA template strand. We're going to use that to make our messenger RNA copy. The job of messenger RNA is to carry the message from the nucleus where the DNA is stored to the ribosome, whether that's attached or it's a free-floating ribosome, and it's going to make a protein at that ribosome. So hopefully by the end of this hour, you'll understand what that little graphic means. All right, a lot of stuff here, a lot of stuff. You see basically from this that there's a ton of standards that we have to cover with all this. And by no means am I going to go through and read all of them, but I will look at some of them. So we know genetic information has to flow from DNA to a messenger RNA, and then it goes to that ribosome to make a protein. So in order for a protein to be made, we have to get the information from the DNA in the nucleus to the RNA so it can be sent out of the nucleus to the ribosome. That's what we know. All right. What enzyme does that is RNA polymerase. So let's see how RNA polymerase directs the making of a DNA into a messenger RNA molecule. That process we know is known as transcription. All right, so let's see how that works. So first we have to tell that RNA polymerase where to go. RNA polymerase is an enzyme that is going to copy DNA, or technically, let's say not copy it, let's say transcribe DNA into RNA. So here's the initiation of how it works. So we're going to break this up into 3 steps. Initiation is the first one. So this is my strand that I want to actually make into a protein. So this is what I'm going to have to transcribe right here. Well, upstream from that just basically means before that is a fancy way to say it. But in all the curriculum and all the questions, or most of the questions, you're going to see upstream. So upstream is a fancy way, again, to say before my strand I need to transcribe. So upstream, I have a promoter. A promoter, usually think about what a promoter is. It's something that's going to get something started or get something going. If you're promoting a product, you want to sell it. You want to get it going. You want people to buy it. So a promoter of a gene is something that's going to get transcription started. So on that, we have a TATA box. That's at the beginning of the promoter. And that's the location where we're going to get transcription factors to bind. Those transcription factors are going to bind. And this is kind of like a signaling sequence. We have a TATA box, the transcription factors bind there. When we have transcription factors, we get this little RNA polymerase to bind, and that RNA polymerase is what's going to make my RNA transcript. So these two words are interchangeable. Messenger RNA and a transcript are usually very interchangeable. You can see both on questions. So once that binds, once the RNA binds where the transcription factors bound on the TATA box, then we initiate transcription. Okay, so let's come up to this big infographic. So the RNA polymerase is bound. We have 2 strands of DNA. You're saying, which one do we use? Well, we're going to use something called the template strand. Okay. The template strand is what we're going to copy our messenger RNA from. And once we have bound that, we just go. That's called elongation. So we're making that transcript until it gets to the end, and that's termination. Okay. So a very simplistic way for a semi-complicated process. So transcription factors bind to the TATA box. That's where the polymerase binds, and then we transcribe our messenger RNA. We use the template strand. Okay, let's come over here and see what it says about the template strand. So the template strand is also referred to the non-coding strand. It's the non-coding strand because we are not coding that strand. We're using as a template for our code. So the template strand is also referred to the non-coding. Template strand is referred to as the non-coding. The non-template strand is referred to the coding strand because your proteins will actually match up to that a little bit better than the template strand because that's the code we use. Okay. So once we're done, we have a completed transcript. What do we do from there? What do we do from there? So we've initiated, TATA box on the promoter, transcription factors are bound, the polymerase is bound. That polymerase reads the DNA template strand to make me an RNA transcript. Okay. So what do we get from here? We get a question to test you on, did you understand any of what I just said? So which of the following explains what we looked at in this process here? All right, I'll give you a second to read it while I pull up your polling feature. And I'll give you about 45 more seconds here. So use some elimination here. Use some elimination skills and think about what we've talked about, maybe what we haven't talked about. So what are we doing? What are we doing? What process is happening here, in other words? Okay, we'll close this down in about 10 seconds. You're going to want to take about a minute per multiple choice, maybe a minute and 10, 15 seconds. But otherwise, you're going to start really cutting into your time and be rushed for time on the exam. All right, so our results say B. Our results say B. We only got about 12 people answering the question there, 13. So is it replication? Maybe. Transcription? Maybe. I can automatically eliminate C and D. Here's why. Just based on the course of what we're doing, translation, we haven't even talked about yet. Alternative splicing? No, haven't talked about it. So based on how this has kind of been organized so far, if we haven't talked about it, we probably haven't had a question about it. So not translation. Translation has nothing to do with the DNA that we just showed. Alternative splicing, I'll talk about in just a second. So it's either transcription or replication. We've talked about both of those. How do we know we are not replicating the DNA, but we are transcribing? How do we know that we are not how do we know we are transcribing and not replicating? It is, somebody threw an answer in the chat. Let's see. Yeah, that would be the U there, right? So the U or uracil is only in RNA. It's not in DNA. So if we were replicating the DNA, we would see T's there and not U's. So that's your biggest clue. Remember, that's a difference between DNA and RNA. That RNA is also single-stranded, which we're only making one strand. Okay, so very good there. Very good. All right, so let's move on to the next step, and that is RNA processing. So I copied that last block here. So we actually have a section of stuff to do before we can move out of the nucleus. Basically, we are just getting it ready, editing. So the pre-mRNA is kind of like a rough draft before we actually go to the final draft. All right. So we've got to do about 3 or 4 things. Okay. And here's what those 3 or 4 things are. We have to add a 5' cap. So we do a cap at the beginning on the 5' end. So we're putting a cap on it. We're putting a tail on it. That tail is a poly A tail. So that's just a bunch of A's, just a bunch of A bases that we would put at the end. Okay. You're saying, well, what's the purpose of putting a cap and a tail on there? Well, this is the protected information right here. So that's where we're going to start. That's where we're going to stop. So this is called the protein coding section. That is what I want to use as a code to make my protein. So if I'm going out of the nucleus into the ribosome, I'm going into a more harsh environment. So if something happened and I chopped up some of this information right here, well, it's not really hurting the protein coding section because it's just a protected cap and a protected tail that I put on it to protect that information. All right. So that's why I add the cap and the tail, is to protect that protein information there. Okay. So you also see a UTR. This is the untranslated region. So that's the area before the start. And then you also have an untranslated region on this side. That's the area after the stop. So you know that we start at the start and we stop at the stop when we get to translation. So we have information before that, information after that, to kind of protect the important information. All right. So that's adding the cap, that's adding the tail. Now we have to look at what these introns and exons are. All right. So this is the way to remember this. Exons start with an X. So that's what we need to express. They don't start with an X, they start with an EX, but we need to express them. Introns start with the IN there. So they interrupt. Okay. So what do they interrupt? They interrupt what we want to code for. So that means we need to get introns out. How do we get them out? By this process called splicing. All right, so you see the introns there in the middle of the 2 exons. So we use these things called spliceosomes. What are spliceosomes made of? Basically, they're scissors that are going to cut out the introns or the interrupting sequence. They're made out of proteins and SNRPs. Okay, so those SNRPs and proteins come together to make a spliceosome, which is a fancy scissor that just pushes out that intron section. And as it pushes out that intron, the exons are spliced together. So this is called exon splicing, because we're cutting out the introns and putting together the exons. So at the end, in the final copy, or the mature RNA or the mature transcript, this is pre-mRNA, this is post-mRNA, or final or mature, whatever you want to say. Those are all kind of interchangeable. We get a 5' cap, a poly A tail. We only have exons. We have a little bit of untranslated on each side, but most importantly, we have our translatable region right here. Okay, and that is what you know as RNA editing, or processing would be another way to say that. Okay, we'll see what we come up with here. It happens differently. It happens differently in prokaryotes and eukaryotes. You know this is a bacterial or a prokaryotic cell because I do not see a nucleus here. Okay. Over here, there is a nucleus, so clearly that's a eukaryotic cell. The difference is bacterial cells don't go through editing. There's not a pre-RNA. It goes straight from DNA to the transcript. In eukaryotic cells, we do have that editing process. So there's an extra step there in eukaryotes. And also it leaves the nucleus in eukaryotes, where it goes straight to the ribosome here in the bacterial cell. We'll look at that picture again after translation. All right, so a couple of questions about that, that editing process. I'll let you look at those before we go on to actual translation. Sorry. There you go. So which of the following explains what's happening in figure 1? So it looks like I've got all those things that I just talked about. All those things that I just talked about. I see some UTRs, I see some exons, I see some introns, I see a poly A tail at some point. See a 5' cap. So think about what all that stuff is going from line 1 to line 2 to line 3. Ten seconds. Okay, let's end that. Okay. So the best answer there, the best answer says that this is the process of editing, right? So this is our pre-mRNA. It doesn't have a cap. It doesn't have a tail on it. It just has the untranslated regions on each side, and it has the introns mixed up in the exons. So if we take out the introns, that is going to allow those exons to come together. So that's exon splicing. I put a 5 cap and a poly A tail on there. This is my mature or final or processed messenger RNA. So that has to be D, right? This is a process of making pre-mRNA into mature RNA. Okay. Again, we haven't translated it all. We haven't modified any protein function. And this is not the correct answer because it's already been synthesized, and it was synthesized just like we made DNA. It was synthesized at the 3' end in a 5 to 3 direction, but that's not going from this step to this step. The synthesis actually occurred here. It's asking what happened from here to here. Okay. So here's another question. Let's see if we can use what we just learned to answer that. I'll launch the poll after you read. So which of the following describes step 2? So if we go all the way back, this is the central dogma again, with step 2 having a little bit of extra in it. All right. Appreciate those of you hanging in there. We've got close to 10 minutes left, maybe a little bit more than 10 minutes. So I appreciate you hanging in there, participating. I know your teachers are loving the fact that you're answering questions and making yourself better on a Saturday morning. Right, about half of you answered, so I'll give you 10 seconds left to finish that up. If you notice what I've done again, I've kind of asked you another question just like the one we had. Okay. 5, 4, 3, 2, 1 in that one. Okay. So it looks like we've got answer choice D pop up as the most answered. So DNA to RNA to protein, that's the central dogma. That's a process of protein synthesis. That's a process of gene expression, expressing that gene in the DNA to a protein. What does step 2 specifically do, though? Step 2 specifically takes out those non-coding sequences. Those non-coding sequences are introns. Remember, introns interrupt. Exons are what we need to express. So those non-coding sequences are removed, and we have just exons that are allowed to code. If those exons are allowed to code, then we can go do translation. So let's do translation after this one question that we have here. So This is a two-parter. So let's do the first part. Let's do the first part. Do not worry about the last part. Just listen to me. Okay. I want you to do the describe. I want you to do the describe and identify. Private message. So we have time to go on. So describe the modification that most likely results in it going from 15 to 7, so the 8 kb difference in the mature RNA, and then identify the location where that 8 difference occurs. You don't even have to make it complete sentences. You don't have to make it complete sentences. Private message me, please. Curtis, very good. All right, good. So what's happening to get me a difference? I've got 2 answers in. I need several more, please. Okay, good. Give you about 30 more seconds here. Okay, I'm gonna cut that one off here. So this is what's happening. Obviously, we're going from here to here, and then the mature RNA is what comes out. So the difference going from 15 long to 7 long has to be the fact that we took out the introns. So the introns are removed, the spliceosomes take out those introns, and this occurs in the nucleus, so we can send out that mature RNA to make a protein. Okay, so let's see if we can do this very quickly. What would be the length if it happened in a prokaryote? What would be the length if it happened? What would be the length of the messenger RNA if it happened in a prokaryote? What would be the length if it happened in a prokaryote? Just give me a number. So I'm not asking you to explain this one or justify like it says, but just give me the number of what it would be if it happened in a prokaryote. How long would my transcript be if it happened in a prokaryote? Okay. Thank you for your answers. The answer would be that it would be 15. Okay, here's why that's 15. So this would be what you would write for your justification. It would be 15 because there's no editing in a prokaryote. Okay, remember it goes straight from DNA to the RNA and then to a protein. So there's no editing. There's no editing. So there would be no change in length. It would be 15. Okay. So yeah, no modification occurs in a prokaryote, only in eukaryote. All right. So we are going to deal with 3 types of RNA. We have seen messenger RNA already. So messenger RNA is, is this red strip right here. It's going to carry the message from the nucleus to the ribosome. Transfer RNA is tRNA. So it carries the anticodon that matches up with the code. So messenger RNA is broken into 3 nucleotide sets. Those are called codons. Those codons match up with the anticodon that will be on the tRNA. So this is what that tRNA looks like. It looks like a T kind of, so you remember it that way. And that's going to have an amino acid up here. So it's going to carry that amino acid. The code anticodon matches with the codon, and up here it's going to have a specific amino acid to bring to the party, if you will. All that happens at the ribosomal RNA. The ribosomal RNA has 2 parts. It has a small and a large subunit. Those numbers differ, 30, 50, 40, 60, depending on if you're in a prokaryote or a eukaryote. Okay, so let's look at how all that works as we finish up with translation. So translation is going to be all of these boxes, which looks like a lot, obviously, but basically it involves moving that ultimate messenger RNA message into a amino acid sequence. We connect those together by peptide bonds, and then we fold them and we get a functional protein. Okay, so for time's sake, let's just look at the next slide and see what we've got here. All of it's on here, so I know this is a lot for you to take in a short amount of space. Everything we've done today, whether that's DNA replication, whether that's transcription, whether that's translation, all occurs in 3 steps. Those 3 steps are always the same: initiation, elongation, and termination. So initiation happens when we get to the start codon. So there's only one start codon, that's AUG. This is a base pair table. This is a codon table. Sometimes you see it as a circle. I just threw that in there so you know what it is. So when we get an A followed by a U followed by a G there, that's AUG. That is my start codon, and that initiates everything to happen. So this is initiation. All right, so start comes in, we get the small subunit with the large subunit, it binds, and so every single protein that's made, every single one of them, will always start with methionine, the amino acid methionine. Okay, that's because that's what the start sequence codons brings in. So this is the codon, this is the anticodon, this is the tRNA, that's the amino acid. So this is the rRNA that comes together, and that initiates translation. So this is what we call the translation initiation complex. And then it's really simple from there, to be honest with you. So what's going to come into this A site? Whatever matches up, whatever anticodon matches up with that codon. And we're going to keep going, and we're going to keep going, and we're going to keep going until we get to the stop. So you see that process happen here. So we have one in the P site, one comes in the A site. We just scoot it over, attach those amino acids with a peptide bond, kick that one out. The E stands for exit, so it exits. That one scoots over from the A site to the P site. A new one comes in the A site, and it brings in whatever amino acid matches up to that codon, and we just keep going over and over. Okay, we keep going over and over until we reach one of these 3 stops. So stop, stop, stop is UAA, UAG, or UGA. When we get there, guess what? It all falls apart. That signifies that we are at the end of our protein and protein synthesis is complete. That free polypeptide, called a polypeptide because there's a bunch of peptides there connecting all those amino acids together, it folds up into its conformational shape. And then if it's an enzyme, it's got it has an active site that substrate can come into. There's a ton of different functions for proteins. It could be a structural protein. It could be a membrane protein. It could be a little bit of everything, and it's going to go do its job once it's there. This is a disposable copy. That messenger RNA is a disposable copy. It can be used again, but a lot of times it's disposed of, and if it gets messed up, we can just make another one from the DNA that we have in the nucleus. Okay. So here is a summary of everything. You've got your initiation, you got your elongation, you got your termination on all 3. So let's go back and review real quick and finish up. So this is my template strand. I use that to make my messenger RNA. Remember, I cut out my introns, I spliced together my exons, I put a poly A tail and a 5' cap on it. That's what comes out of that nuclear pore, and it goes to the ribosome. Okay. We start reading that when we get to the start codon, and then we add different amino acids based on the codon anticodon pair. And whenever we get to the end, we terminate that, and that makes my protein. So hopefully transcription and translation, you at least have kind of seen it. So when we go to it, you're going to understand it when we start unit 6. Okay. Remember, it happens differently in prokaryotes and eukaryotes. This is prokaryotic. So since there's no RNA editing, I can immediately send it to the ribosome. So technically, transcription and translation happen simultaneously in a eukaryote, but it cannot do that in a prokaryote because we have to edit it and send it out of the nucleus. Okay. So let's finish with these 2 questions here, and we will gotten through everything today. So the mRNA transcribed from the DNA would read what? So I've got this right here as the strand. What would match up to that? So this is the DNA. What RNA would match up to that? I will launch the poll. I will launch the poll here for you to give me the last 2 questions. I've got one more after this one. More than likely, you're not going to have to look at all of it. It'll probably give you the answer pretty quickly. There's a clue in there to eliminate a couple of those very quickly. And nobody has picked those yet. So very good. About 10 seconds to close this poll. Okay. So I'm going to close that in 5, 4, 3, 2, 1. Okay. So nobody took the bait on A or B. Remember, we're looking for mRNA, so it can't have T's in it. That would be DNA. So it would be either this one or this one. And the clue is this one's easy to identify. AAA would have to be UUU, so it's automatically C because that doesn't match up to that third triplet. Do we use this box up here at all? No, we only use that when we start to figure out what is translated, not transcribed. So I thought this was interesting that they put that in the question. It was just kind of to trick you maybe in what you were doing. Okay, finish up with this question and then we'll call it a day. All right. I'll give you a second and we'll launch that after you had a second to read it. So we talked about the difference in prokaryotes and eukaryotes a few times. Okay, about 15 seconds. We're a little all over the place on this one. Okay. So the correct answer on this one, the processes will occur simultaneously in prokaryotes. Remember, they happen simultaneously because the transcription and translation both happen in the cytoplasm, but that happens in prokaryotes and it can't happen in eukaryotes because there's 2 different locations for transcription and translation. So the right answer is A. And that will finish us up for the day. So thanks for everybody coming in. Our next study session is going to be a month from today, on March 6th. So we'll see you again there. I'll hang out just a minute. If anybody has any questions, feel free to throw those in the chat box. Hopefully this is beneficial. You can reach out. My email was on the front slide. Let me know if you have any questions and I'll be happy to answer them. So good luck. We'll see you in a month.